I want to just start with a quick review of what we've discussed last class. And this is what we're going to rely heavily on in our today's discussion. So first, we have discussed the structure of the heart. We stated that the heart is formed of 4 chambers. We have right atrium, right ventricle, left atrium, and left ventricle. And we mentioned that the atria are connected to veins regardless of the state of oxygenation. So on the right side, the right atrium is going to be receiving the deoxygenated blood through 3 major veins: superior vena cava, inferior vena cava, and the coronary sinus that drains the heart itself. The oxygenated blood returning from the lungs will be drained from the lungs into the left atrium through the pulmonary veins. Remember that connected to the ventricles, we don't have veins, we have arteries. Right ventricle is going to be pumping the deoxygenated blood to travel to the lungs for it to become oxygenated through the pulmonary trunk that will split into right and left pulmonary arteries. And the oxygenated blood is going to be pumped from the left ventricle through the largest systemic artery, which is my aorta. That will be distributing the oxygenated blood to all the different organs, all the different systemic organs. Remember, the blood flows in this pattern. So it travels from a systemic organ through veins, either superior or inferior vena cava to the right atrium. And then from the right atrium, it travels down to the right ventricle. What allows this unidirectional flow between the right atrium and right ventricle is a valve that we call the tricuspid valve. And if you remember, this is one of the 2 AV valves that we have. When the ventricles contract, what they will do, the right ventricle pumps the deoxygenated blood to travel towards the lungs, passing through a valve located at the base of the pulmonary trunk. We call this is my pulmonary semilunar valve. This deoxygenated blood, when it reaches the lungs will allow the oxygenation to take place. And now the blood becomes oxygenated and would be drained from the lungs towards my heart through the pulmonary veins. Remember, this oxygenated blood that traveled to the left atrium will travel down to the left ventricle, passing through another AV valve. This is my mitral valve, or what we call the bicuspid valve. Blood will then be pumped from the ventricles. The blood from the left side, from the left ventricle, is going to be pumped out to travel into the aorta to be distributed to the systemic organs, passing through the aortic semilunar valve. This oxygenated blood will find its way to the various systemic organs, and those organs will be consuming the oxygen, and the blood now becomes deoxygenated, which is going to be drained again through superior inferior vena cava to the right atrium, to the right ventricle, to the lungs, back from the lungs to left atrium, to the left ventricle, and then to the aorta, and then to the other systemic organs, and so on. All right, so those are 2 combined circuits, if you remember. those two combined circuits here, those are the pulmonary and the systemic circuits. All right. What else did we discuss last lecture? We discussed the skeletal muscle contraction. If you remember, skeletal muscles will need to receive an electrical signal from the brain, from the primary motor cortex. And this electrical signal will be in the form of an action potential traveling along the length of my nerve cell until it reaches the terminal nodes of my neurons. When the electrical signal reaches the terminal node of my neuron, this will open, if you remember, voltage gated calcium channels, allowing the calcium to pass to the inside of the terminal end of the nerve cell. This influx of calcium, if you remember, did stimulate the vesicles storing the neurotransmitter to fuse with the cell membrane and to release the neurotransmitter to the outside of the nerve cell. And we call this neurotransmitter in the neuromuscular junctions, this is my acetylcholine. How the acetylcholine did affect the muscle for it to contract. Remember, the acetylcholine binds to its receptor, and the receptor associated with ion channels. Remember, in a resting membrane potential, when I don't have a signal, the inside of the cell is more negatively charged than the outside of the cell, which is going to be more positively charged. When the acetylcholine binds to its receptor, what's going to happen? This will open sodium channels. So when I get the acetylcholine attached to its receptor, I open sodium channels, and this will allow the sodium into the cell, changing the membrane polarity. So now the inside becomes more positively charged compared to the outside, which will become more negatively charged. So I've created another electrical signal. This time is traveling along my cytoplasmic membrane of the muscle cell. Remember, this electrical stimulus traveling along the length of the muscle cell membrane is going to be passing to the inside of the cell, traveling through T tubules that are in direct contact with flattened tubules located inside the cells. So this is what we've called the sarcoplasmic reticulum. Remember, what was the effect of this electrical signal? It opened another calcium channel, another voltage gated calcium channel, allowing calcium to travel to the inside of the cell. This time is a muscle cell. And this calcium passing to the inside will stimulate the sarcoplasmic reticulum, the smooth endoplasmic reticulum of the skeletal muscle cell, to release calcium to the inside now of the cytoplasm of the muscle cell. So all this is inside the muscle cell. How the calcium did allow the contraction to take place. Remember, inside the muscles, we do have contractile filaments. And those contractile filaments are going to be either thick filaments or thin filaments. Thick filaments are formed by protein subunits called the myosin. And the thin filaments are formed by protein subunits called actin. Remember that not only the actin is going to be forming my thin filaments, I have regulatory proteins as well that will be forming my thin filaments, and those are the troponin and the tropomyosin. So what happens when I release this calcium in the cytoplasm? This calcium now gets in contact with my troponin. When it binds to the troponin changes its shape. And as it changes its shape, what's going to happen? It's going to be moving the tropomyosin away, like this, exposing the binding site on the actin subunit. So now I have exposed the binding site on the actin. This is my binding site that appeared on the actin. Once I expose the binding site on the actin subunits, the myosin will form cross bridges that will bind to the actin and will be dragging the actin, in other words, the thin filaments, to slide along the length of the thick filaments. And this will do what to the length of the muscle cell, or the muscle fiber here. This is during relaxation. When I keep sliding those tiny filaments inside, this will cause the shortening in the length of my muscle fibers. As every single muscle fiber shortens, what's going to happen to the whole length of the muscle? The whole skeletal muscle going to be shortened, allowing the contraction to take place. This is how the skeletal muscle did take place. The skeletal muscle contraction did take place. Remember, the cardiac muscle contracts in the same exact way with few differences. So everything that we've mentioned here will be applied except for first thing, I don't need an external electrical impulse to initiate the contraction. So I don't wait for the brain to send me an electrical signal to initiate my muscle contraction. I have 1% of the cardiac muscle cells that can generate their own electrical impulses. They have automaticity. 1% of the cardiac muscle cells are going to be sending out the electrical impulse. Another major difference between skeletal muscle contraction and cardiac muscle contraction is that it's not essentially, in skeletal muscle contractions, it's not going to be essential to send out the electrical signal to every single muscle cell in the skeletal muscle every time you decide to use the muscle. So when I flex my forearm, when I contract my biceps, I don't have to use all the muscle fibers in this biceps for me to perform any contraction. The grade of the muscle contraction is going to be determined according to the load that you are trying to move. But on the other hand, cardiac muscle are going to be connected all together by gap junctions. And what's a gap junction? If you remember, a gap junction is an attachment between the cells in which I have channels connecting the cells together. It's an attachment between the cells, but I have channels. So what does this mean for me? This means that when you get sodium inside one cell, this sodium can freely travel to all the rest of the cells. So this means what? This means that once I get a signal to one cell, it will automatically be transferred to all the cardiac muscle cells. This assures what? This assures that all the cardiac muscle cells gonna be contracting in response to the signal. All right. We also did discuss this 1% of the cardiac muscle cells that have automaticity. And if you remember, those were a node located in the right atrium called the sinoatrial node or SA node. Another node that will be located between the atrium and the ventricle. And we've got embedded fibers or cardiac cells that are going to be embedded in the wall of the ventricles. So what do we call the one at the junction between the atrium and the ventricle? We call it the atrioventricular node or the AV node. And please don't confuse AV node with the AV valves. A valve is a connective tissue that prevents the backflow of the blood. This is AV valve. This is either mitral valve or tricuspid valve. This is a valve. This is a connective tissue that keeps moving up and down according to the difference in the pressure of the blood between the two sides. But here, AV node is a cluster of cardiac muscle cells that have the ability to generate electrical impulses. And the ones that will be located embedded in the wall of the ventricles, those are what we've called the Purkinje fibers. Remember, which one would I follow? SA node generates 75 depolarizations per minute on average. Do you remember how many depolarizations can be generated per minute by the AV node? 50 depolarizations, exactly. How on average? And how about the Purkinje fibers? 30 depolarizations. So which one would I follow? You follow the fastest node, and the fastest node normally is my SA node. What if, for any reason, another part of the heart did start to generate electrical impulses faster than the SA node? Which one would the heart follow? The pathological node or the SA node? If, for example, I am firing 100 depolarizations per minute, which one would I follow? The pathological node or the SA node? SA, 100, SA, what do you think? Which one? Which one would you follow? Your cardiac muscles follow. The fastest node, which one is the fastest node in this case? The pathological one, this one on here. That's why I will have disturbance in my heart rate. Ectopic focus. All right, so you follow the fastest node, regardless if it's normal, a normal node that you have in the heart or a pathological node. All right. So we're looking in here at how the electrical impulses will be traveling as they get generated from the SA node, which I normally follow. Remember, cardiac muscle cells are connected by gap junctions. So what's going to happen to this electrical impulse that was generated from the SA node? It freely travels to all the cardiac muscle cells of the atria, right and left atria. So both atria will be depolarized and then they will be repolarized. Why not the ventricles at the same time? Because the ventricles are connected to the atria electrically only through the AV node and AV bundle. This is my AV bundle on here. So what is the problem here with the AV node? The AV node has fewer gap junctions compared to the rest of the heart. So there will be some time delay of 0.1 second while the electrical impulse is going to be freely distributed to the cardiac muscle cells of the atria. It reaches the AV node, which is the only port to leave the atria and get into the ventricles. But this AV node will be delaying the electrical conduction. What happens next? The electrical impulse will travel from the AV node and AV bundle to the right and left bundle branches, which will be distributing the electrical signal to the Purkinje fibers and the rest of the cardiac muscle cells of the ventricles. The excitation of those cardiac muscle cells of the ventricles starts from the apex upwards, like this, in this direction. So I started with an SA node firing depolarizations, an average 75 depolarizations per minute, traveling freely across the cardiac muscle cells of the atria. And then as I'm moving down to the ventricles, this electrical signal gets delayed, passing through the AV node. So if you remember, what was the action potentials, the regular action potentials that we've studied back in the nervous system in AP1? If you remember, an action potential starts with a depolarization phase. If this is a nerve fiber, here I'm looking back at what we've discussed in AP1. Remember, during rest, the inside of the cell membrane is more negatively charged than the outside. This is during the resting membrane potential stage. What happens if I received a threshold stimulus? If I received a threshold stimulus, this will cause the opening of sodium channels, special type of sodium channels. Don't want to get into this detail. As I allow the sodium in, what's going to happen? Inside will become more positively charged as the sodium is more positively charged, is positive. And outside will become more negatively charged. Here we're checking the difference between the inside and outside. I'm not saying that inside is purely negatively charged or outside is purely positively charged. It's a matter of comparison here. I'm comparing inside and outside. So I allow the sodium in, inside will become more positively charged. And then in order for this cell to respond to another stimulus, it needs to return back to its resting membrane potential. I need to get back for the inside to become negatively charged, and the outside to return back to become more positively charged. So what will I do? As a major cation inside the cell is the potassium, we open the potassium channels, allowing the potassium to flow outside of the cell. So losing positively charged ions will allow you to return to the polarized state. If you remember from last time, potassium is responsible for the polarized state. It allows the cell to get back to the polarized state. What I mean by polarized state means the inside is positive, is the outside, the inside is more negatively charged than the outside. All right. So looking here, if you remember also, those potassium channels that you did open for you to get back to the polarized state in a phase called repolarization was leaking potassium even beyond your resting membrane potential. So even after I reach the resting membrane potential, I will still be leaking some potassium out. So the inside won't only become negatively charged as it was, but it will even become more negatively charged than the resting membrane potential. So here I was 1 negative on the inside, now I have 2 negative on the inside. So means what? Means I have a more negatively charged membrane on the inside than it was before I sent the electrical signal. So we call this is an exaggerated polarity. What do we call this exaggerated polarity? Is hyper-polarization. All right. So what are the phases in here of an action potential that we've studied back in AP1 in the nervous system? We started by a resting membrane potential. Inside is more negative, outside is more positive. Allowed sodium in, you've lost the polarity. We call this phase depolarization phase. I have lost the polarity. Then I get back the polarity by allowing potassium out. This is my repolarization. I return back to the polarized state. But those potassium channels will keep leaking potassium to the outside of the cell even after I reach the resting membrane potential. So what do we call this exaggerated polarity? It's hyperpolarization. So if I'm drawing this on a graph on here, if I'm starting, let's say, as a nerve, negative 70 millivolts. So the first phase, inside becomes more positive. So here I was negative 70 millivolts. So I become more positive, means the graph is going up or down. What do you think? This is my 0 on here. Will I be going up or down? From this point, my resting membrane potential, I'm going up. This is my first phase, depolarization. So now I've completed the delivery of this electrical signal. I've reached a certain level here at which we're going to close the sodium and I open the potassium. So now I am losing potassium out. So this means I will become more negatively charged or more positively charged on the inside. What we're concerned with is inside. When you leak potassium, when you get potassium out of the cell, you're going to become more negative or more positive? more negative. So you see on here, you're going to become more negatively charged now. So you're going to be moving down until my resting membrane potential here. This is what we call my repolarization. But if you remember, the potassium kept leaking outside of the cell, even after you reach the resting membrane potential that you started with, even after I reached negative 70 millivolts. So what's going to happen? I will become even more negatively charged than I was before conducting the signal. We call this phase as hyperpolarization. All right, so we have three main phases, or four main phases actually. We started by resting membrane, depolarization, repolarization, and the exaggerated polarity here. This is hyperpolarization. If you remember, during depolarization, what channels are open? What is the channels that is open? That allows the depolarization to take place? Sodium, right? So sodium channels are open. During early repolarization, what am I doing? First thing, I closed the sodium channels. And then I opened, and in the same time I opened the potassium channels to allow the potassium out. So two things will take place. I close sodium and open potassium. Here, what channels are still open during hyperpolarization? I still have potassium channels leaking potassium out. So what if I am getting another signal here during depolarization? So I started the depolarization. I've got another signal. How was the nerve responding to this other signal if it received another signal during depolarization? What do you think? Would I respond? What do you think? Needs a higher stimulus? Even a higher stimulus. What do you think? So this is a stimulus that initiated the action potential. And during depolarization, I got a much stronger stimulus. What's going to happen? During depolarization, what do you think? Nothing going to happen. Why? Because the sodium channel is open. So I can't open an open door. If the door is already open, you can't open it while it's open. It's already open. This is how I respond. I open the door. If the door is already opened, so can you open it while it's opened? You can't. So what's going to happen in response to this higher stimulus? Nothing. How about here? While I'm closing during early repolarization, you've got the same very strong stimulus. What do you think? You've got the same very strong stimulus. During early repolarization, again, what am I doing? During early repolarization, I am closing the sodium channels. What's going to be the response here in this case? If you got a stimulus to the nerve while it's closing the sodium, Would I start here to create another electrical signal to be sent? What do you think? If I'm closing the sodium channels during early repolarization, what do you think? Any other guesses? How do you respond again? Can you remind me? You respond by opening sodium channels, right? If I'm telling you that during this phase, you are closing the sodium channels, can you open and close a door both in the same time? Can you open and close it in the same time? No. So what will happen in response to this stimulus? Again, nothing. So this time, during depolarization and early repolarization, all this period here, I won't be able to respond to any kind of stimulus, even a very strong stimulus. I can't respond. We call this period is the absolute refractory period. What is absolute refractory period means? This is a period you want to be responding to any stimulus, whatever is the strength of the stimulus, strong, threshold, whatever. How about I've already closed the sodium channels during late repolarization. What is open now, it's the potassium channels. During late repolarization and during hyperpolarization, what's open now are the potassium channels. And I'm leaking potassium out here. What if I got a stimulus which is strong during this phase that won't only get the needed number of sodium channels to be opened to create an action potential, but also to open enough sodium channels to get sodium to substitute for the potassium I'm leaking out. What do you think? Would I be able to initiate another action potential in this case if I received a strong stimulus that can compensate for the loss of potassium in the same time, open enough sodium channels to create another depolarization phase? What do you think? Yes, in this case, I can create another action potential. But if I got a strong stimulus, what if you got the minimal stimulus? You will not be able to compensate for the potassium that you're leaking here. So what's going to happen if you get a threshold stimulus during late repolarization and during hyperpolarization? You will not respond. It requires you to get a strong stimulus, stronger than threshold. So here, what do we call this phase during late repolarization and during hyperpolarization? This is my relative refractory period. What's relative refractory period? It's a refractory period that is relative. What I mean by relative, it's relative to the strength of the stimulus. I will respond to a strong stimulus, but if you give me a threshold stimulus, I will not respond. So again, what is absolute refractory period? It means the period of time during which the nerve or excitable cell will not respond. And this here, in case of the nerve, it will not be responding during depolarization and early repolarization. How about relative refractory period? The nerve cell will not respond to create another signal unless it receives a strong stimulus. This is a relative refractory period. Any questions? Any questions? Questions? All right, how about the cardiac muscle cells? How the action potential of the cardiac muscle cell looks like. Very first phase of the action potential of the cardiac muscle cell is going to be due to the opening of sodium channels, the same like what we've seen in the nerve. So I allow the sodium in, sodium channels are open, inside will become more positively charged, so I'm going up. The same exact thing that takes place in the nerve cell. After that, after you depolarize, what you're gonna do? You're gonna be doing what here? After you depolarize, what you're gonna do next? You repolarize. And how would you repolarize? Again, you close sodium channels and you open potassium channels. But can you remind me the last things that we've mentioned before we leave in our last meeting? What was the response of a muscle to a depolarization? If the muscle gets depolarized, how does it respond? Yeah, I see two answers now. Any other? If I depolarize, the muscle will contract. How about during repolarization? When I repolarize, I do what? I relax. Exactly. Remember, is this something good for the heart to rapidly contract? Contract very, 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 very fast. Why not? Can you remind me why it's not something good to keep contracting the heart very fast? It needs to relax to do what? To refill. Exactly. So during contraction, I push the blood out. But during relaxation, I fill the chamber with blood. All right, so I need to relax. What if here I got a signal, another stimulus in here to the cardiac muscle cell? It should be during late repolarization here. If it was the same as the nerve, it will be during late repolarization. So if I received a strong stimulus, what's going to happen? If it was like the nerve, what's going to happen? It will be creating another action potential, and so another depolarization, which causes it to contract. Do I want this? Or would I want to assure that the cardiac muscles got enough time for them to relax? I need to ensure here that the cardiac muscle cells got enough time to relax. So I want to do something like this. This is my early repolarization phase. That I want something that will delay the repolarization, something that will make the graph here to become parallel to the x-axis. What I mean by a line that is parallel to the x-axis in a graph, what this means, that this is constant, and here, what is this here? It's the voltage. And on the x-axis, this is not constant. And here on the graph, it's time. So what I mean by having this flat line on the graph, this simply means that there is no change in the voltage, but there is time passing. I'm delaying the repolarization. All right, so how would I do this? How would I maintain a constant voltage in the cardiac muscle cells? Again, what happened in here? I did allow the sodium in, allowing me to depolarize first, and then I close the sodium and open the potassium for me to repolarize. So I'm getting potassium out. If I want you to keep the voltage constant, and I'm telling you here, you are doing what? You are releasing potassium out of the cell. So what should you do in this case? If I'm asking you to keep the voltage of the cardiac muscle cell constant, what would you do? Close potassium, but remember, it's so hard to close the potassium. Even here in the nerve, they are kept leaking potassium out. And there is no way I can close it and reopen it again. So can you give me another way by which I can deal with this? What do you think? Any other ideas? Not interfering with potassium, because you did already open the potassium channels. You want actually to repolarize, but you want just to delay the repolarization by keeping the voltage constant for a while. What do you think is another thing you can do other than limiting the efflux of potassium? The efflux of potassium will keep going. So what else would you do? Let me ask you the question in terms of money. You would know this is better. All right, so for example, here you have $5,000 in a bank account and you're earning $1,000. I told you that you're getting $1,000 into your bank account and you're gonna spend it. So what is gonna be the amount that will remain after you spend this $1,000 in your account? It's gonna be the same. It's gonna be constant, the same. So what do you need here? In terms of potassium, I want to keep the same amount of positively charged ions while I'm spending positive, while I'm losing positive. So I have here in my bank account, I'm telling you now you have 5,000 and you're going to be spending 1,000. what you're going to do to keep this 5,000 constant? You need to do what? To keep the same 5,000? You need to do I'm waiting for an answer. In order to keep the 5,000 bucks constant, and you are spending 1,000 each month, what you're gonna need to do? You need to make 1,000 a month. If you are spending 1,000 a month, in order to make the 5,000 to be constant, you need to make 1,000 each month. All right, is this clear? So here I'm telling you, I'm losing potassium, positive charges, positive charges. I am spending. So what you need to do, you need to gain. I need to gain negative or positively charged ions. What do you think? I'm wasting here potassium. I'm still wasting potassium. But I need to stabilize it. I need the voltage to become constant. Potassium, why the potassium is moving out? Why the potassium is moving out? Potassium has a high concentration inside. So if you kept opening potassium channels, the potassium will keep moving out. So I need another ion other than potassium and other than sodium that exists outside of the cell. And once I open its channels, it can travel in. Other than sodium, because sodium I did just close the channels, and other than potassium because the channels are opened. What is another ion that we've just mentioned earlier today, that once you open a channel for it, it's going to be passing to the inside of the cell. Ion, not acetylcholine. Acetylcholine is going to be a neurotransmitter. We're talking about ions that has a voltage. What is this ion? What is this ion that can travel to the inside of the cell when you open its channels? Calcium. You see on here, so what would I do here to maintain a constant voltage while I'm losing potassium out? To gain positive while I'm losing positive, I need to open calcium channels to get the calcium in. So now the gain is the same as the loss. They are canceling one another. So what's going to happen to my voltage in this phase? It's going to remain constant. We call this is my plateau phase. And why in the heart cells, in the cardiac muscle cells, do I have a plateau phase? To increase the time here during which the cardiac muscle cells are not responding to any stimulus. Why? To make sure that this cardiac muscle cell did have enough time to relax, because you need to relax to refill. So again, what did I do here? What is the major difference between a nerve cell in terms of action potential and a cardiac muscle cell? A cardiac muscle cell has a prolonged repolarization phase. It takes more time during repolarization. And what is the logic here? What is the logic of this great design of having a more prolonged duration for the repolarization to take place? A more prolonged duration for the repolarization to take place is making sure that even if you get other stimuli during this time, the muscle will relax and will not respond to those stimuli. Why would I need to relax? I need to relax because if I have nothing to pump on the next beat, as if I wasn't contracting. So if you contracted your cardiac muscle cells without having enough time to relax, what you will end up having no blood coming out of the heart. So what prolongs, again, the repolarization phase in the cardiac muscle cells is the presence of what phase of the action potential? My plateau phase. And again, what is the ion responsible for allowing the formation of the plateau phase? Again, it's my calcium. All right. Questions? Any questions? So again, what did we see so far is what changed in each cardiac muscle cell. This is the change in electrical voltage of one cell, just one cardiac muscle cell, just one cell. This is the actual potential. This is the electrical change. Last class, we discussed the AKG. And if you remember, AKG or ECG, this is going to be a drawing of the electrical changes, not in one cardiac muscle cell, but in the whole heart. This is an action potential of one cardiac muscle cell. This is just one. The AKGs that we've studied last class, it was thousands of combined action potentials, all taking place in the same time in the heart. So millions and millions of action potentials that will be recorded by the machine as an AKG. If you remember, what was the first thing that was taking place in the heart in terms of electrical change? Signals were created by the normal pacemaker in the heart. This was my SA node. Remember, SA node is a normal pacemaker. It sends out the electrical signals causing the excitation of both the right and left atria. So here, what is the first thing that takes place when you send out a signal? What is the very first thing that happens? What is this phase? This one on here, what do you think? Depolarization. So I depolarize the atria. This is what the machine will be recording. So I'm looking at what in here? I'm looking at the depolarization phase of the action potential of all the cardiac muscle cells of both atria in the same time. This is going to be recorded by the machine as P wave. And then what happened? Remember, this signal can't travel directly to the ventricles through gap junctions. The only connection is going to be by passing through the AV node, which causes some time delay. So I've got some time delay in here. Time delay, no electrical change is going to be recorded. So it's going to be a flat line. No increase, no decrease, nothing. And then what? This electrical signal will find its way to the cardiac muscle cells. of the ventricles of the heart. So this results in what? This results in depolarization of both ventricles at the same time. And remember, this is going to be recorded by the EKG as the QRS complex. So it started by P wave, it's atrial depolarization. And then the same phase of the action potential on here is going to be recorded, but this time of the ventricular cardiac muscle cells. It's going to be recorded as my QRS complex. During this time, while the machine is recording the electrical change taking place in the ventricles, the atria are going to be repolarizing. This is taking place while the ventricles are depolarizing. So that's why it's going to be masked. So I will not be recording the repolarization of the atria. I've recorded depolarization, but when the atria are repolarizing, what's going on in the ventricles are depolarizing. So the ventricles, as they have much thicker wall, they have much greater number of cardiac muscle cells, so they create a much higher voltage. This is the one that's going to be recorded by the EKG. So you've got ventricular depolarization and atrial repolarization taking place in both in the same time. Which one gets recorded is the ventricular depolarization, as you remember from last class. What happens after the ventricles depolarize? They will repolarize. And the ventricular repolarization is going to be recorded by the EKG as my T wave. So what is the repolarization again? All this is going to be my repolarization phase. Recorded first as P wave, recorded first time in the ventricles as my T wave. Exactly. Any questions? Any questions?